cell culture platform that can be used for 3D compartmentalization of primary motor neurons and Schwann cells. The platform consists of 2 median channels on the sides and one extracellular matrix hydrogel channel in the middle, which are separated by PDMA exposed. Motor neurons are suspended through the median channel and then allowed to attach onto the hydrogels. Schwann cells are restricted inside the hydrogel by encapsulation in collagen-1 hydrogels, and the hydrogel channels in the 3D microfluidic platform isolate the Schwann cells and the axons from the soma of the motor neurons. They investigated the role of Schwann cells in axonal outgrowth by comparing motor neurons monoculture and motor neurons and Schwann cells co-cultures. They saw significant outgrowth starting on day 1 of the co-cultures, and the relative axonal growth rate of the monoculture was lower than that of the co-culture. It is important to note that in their platform, the axons grow 5 times faster than on 2D surfaces. So these results reveal that the 3D microenvironment and the presence of the Schwann cells accelerate axonal growth. They compare Schwann cells culture in the hydrogel to those in the 2D cultures to investigate the role of the 3D microenvironment of the Schwann cells in the absence of motor neurons, and in both 3D and 2D cultures, the Schwann cells display 3 morphological phenotypes, and the percentage of each morphological phenotype was similar in 2D and 3D on day 1. However, on day 7, the bipolar phenotype increased in 3D culture. These findings demonstrate that the scaffold, their device, played a critical role in providing a 3D environment that allows a spontaneous transformation of the Schwann cells into the bipolar phenotype, which is similar to the one observed in vitro. They also saw that Schwann cells were aligned with the proximal axons of day 7 of co-culture. However, they had a random distribution on the monoculture. They also investigated the effect of the Schwann cells on neuron maturation. And in this experiment, the Schwann cells are embedded in the collagen inside the hydrogen channel. After 1 week of culture, motor neurons are seated inside the median channel. And on day 7 of the co-culture, ascorbic acid was added to the co-culture medium to induce myelination. And in addition, glial growth factor was supplemented along with the ascorbic acid to develop a demyelination model on day 14 of the co-culture. They saw that Schwann cells played a critical role in neural maturation, as, for example, when expression of NFH is low in developing neurons and increases in large myelinated axons, and they saw that this tendency can be reversed in the demyelination model. Furthermore, when treated with ascorbic acid, the synthesis of collagen type 4 increases in the myelination model, supporting previous findings that this type of collagen serves as a scaffold that attracts Schwann cells to attach to axons and initiate myelination. Terminally differentiated Schwann cells forming compact myelin are known to express MVP, so they further analyzed myelin information by immunolabeling MVP, and they saw that on day 14 of the co-culture, the Schwann cells were mostly in the pre-myelinating stage, with only approximately 30% of cells expressing MVP. However, a higher percentage of Schwann cells expressing MVP was observed on day 21, approximately 75%. Glial growth factor treatment reduced number of myelinating Schwann cells on day 21 to approximately 44%, indicating that maturation and myelination were suppressed in the demyelination model. They demonstrated that supplementing ascorbic acid in the culture medium allowed maturation of the Schwann cells into myelinating cells to initiate the wrapping and formation of the myelin sheath, whereas the glial growth factor treatment of the co-culture inhibited this process. So in conclusion, we propose a robust method. They propose a robust method for modeling peripheral nervous systems myelination using motor neurons and Schwann cells co-culture in a microengineering platform that can be easily used to facilitate co-culture of different types of cells. That would be all. Thank you.
Good morning. Can you clearly see my screen?
We can see,
Yes.
Eric.
Yes.
Thank you for your confirmation. Okay, good morning, everyone. Today I'm going to inform you about predicting anxiety, depression, and stress in modern life using machine learning algorithms. In this paper, prediction of anxiety, depression, and stress were made using several machine learning algorithms because they are highly accurate and appropriate to predicting psychological problems. In this context, the World Health Organization has observed that depression is the most prevalent mental disorder, affecting for more than the 300 million people worldwide. That's why many health researchers to focus their study in this area. Regarding materialism methodology, this research focus on the detecting anxiety, depression, and stress using the depression, anxiety, and stress scala questionnaires. Data were collected from a total of 348 participants, aged between 20 and 60 years, both males and females, employed and unemployed, with a wide range of responsibility, who were asked to complete the questionnaires, subsequently classify using machine learning algorithms, as we will see in the following section. About questionnaires, they comprise 21 questions, with 7 questions allocated to each of the scale of stress, anxiety, and depression, and the possible answer could be given in text or numerical form as follows: 0, 1, 2, 3. For example, 0 didn't apply to me, 1 applied to me some degree. So table 1 provides information about the question asked for individual regarding anxiety, depression, and stress. After the data collection, the responses were encoded using numerical values of 0 to 3, and the scores were then calculated by adding the values associated with each question on the below formulae. Finally, they were labeled according to severity, as shown in table 2. I mean, normal, mild, moderate, severe, and extremely severe. Regarding machine learning algorithms applied in this study, we can mention 5 methods which predict the percentage of people suffering from symptoms of stress, anxiety, and depression according to the level of severity, such as decision 3, which is very useful to predict problems because they are so easy to interpret. The structure is stable, as we can see in the figure 1. A question is divided in yes or no in 2 branches, driving out of the tree. But we also have support vector machines, Bayes, K-Neurus neighbor, that we have analyzed in the previous papers. On the other hand, the application of all 5 methods for all 3 classes, I mean stress, anxiety, and depression, resulted in the confusion matrices as shown in table 3. As you can see here, the rows in the matrices show the actual classes, while the columns shows the predicted classes. The number 1 to 5 in the rows and columns represents normal, mild, moderate, severe, extremely severe cases, as we saw in the table 2. So at the
1
same
minute
time,
left.
okay. At the same time, table 4 shows the high accuracy for all three scales of anxiety, depression, or stress we achieve for need-based. And finally, figure 5 shows the variable importance rating for anxiety, depression, and stress. The higher the number, the more important the variable. I mean, for example, here, the variable scare without any good reason proved to be most important on the anxiety scale, and the variable life when meaningless was found to be important on the depression scale. While difficult to relax was found to be important on the stress scale. As such, these variables were considered to be most important in detecting psychological disorder. That's all. Thank you for your attention.
Okay. My topic: the effects of viscosity on the undulatory swimming dynamics of sea elegans. The swimming dynamics of the Nenato sea elegans with different viscosity was investigated. The organism moves, speeding waves in the agile and efficient weight. In this part, in figure 1a, study the swimming dynamics of the worms is based on the orthogonal segments of the end of the pipette. These were populating the swimming movement of the worm. The rest of the pipette was tilted at 90 grades out of the swimming plane to adjust a force sensing cantilever. And in figure 1b, following the reflection of the micropipette in both directions, lateral drag force and propulsion force were measured directly. In this part, the obtained viscosity of the fluids measurement were made in 5 different solutions: M9, 1%, 5%, 10%, 15%. Here the viscosity is measured as a function of the mass concentration. And this part, kinematic swimming of C. elegans is visually in increasing viscosity in optical microscopy image of the worm during swimming. The swimming curve is illustrated as a function of time, and the coordinate defines the position of each segment of the worm, and the frequency of swimming decrease slightly with the viscosity that is a greater time space between lines on the same curve. And this part, force use time data is observed both for lateral and propellant force. In direction C and D are shown in a low viscosity fluid on the left panels and high on the right panels. Lateral force is 2 times greater than propulsion force, and the peak of the maximum force occurs when the worms move faster to the right and slower to the left. and as the number of Reynolds decrease in fluid of higher viscosity, the nematode phase to a more conventional micronation regimen where viscous force are relevant. And the conclusion, the micropipette technique was used to probe the way swimming of C. elegans with different viscosities, and the worm modulates its gait to increase propulsion without modifying its energy spent. Thanks.
Good morning, everyone. Can you see my screen?
Yes.
Okay, thank you. In this time, I'm going to talk about this paper that is titled Double Emulsion Pretreated Microwell Culture for the In Vitro Production of Multicellular Spheroids and their situ analysis. Okay, for perform this experiment, this title, the objective of this paper is culture spheroids, and they perform the double motion generator. It's a one microfluidic device in order to generate a double motion. One is this performed with medium and oil, and the second with pluronic and medium also. And they use another microfluidic device in order to trap this double motion double motion spheroid, and inside they have a cellular. The line of the cells is a one line that they use for a model, but then we use a long cancer cells in order to get what is the response of the treatment of the drugs of the lung cancer cells. Okay, they perform this experiment with these two chip devices, and they count or they try to trap one cell in this confine in order to study what is the behavior of this. Okay, they use a live cell calcium in order to get what is the viability of the cell and also propedium. And this is the statistics from these experiments. They found a different they found difference after 72 hours of culture. The cells rapidly start to die, and they found this difference and these 72 hours. It means that they only are able to stay in a 48 hours ability. For this reason, they are interested in study the development of these spheroids, and they interrupt the spheroids after 45 hours, and then they study the formation of the spheroid, and they after 7 days they notice that the spheroid after the seven days the spheroid is performing well, and they start to study, but they notice that the polarity of formation it's quite different. In letter D we saw this is the difference between because they use a matrix in order to compare this polarity and they use a Raman spectroscopy in order to find what happened with this polarity, in order to see what is the structure, or what is the polymer structure or the polymer conformation between both a matrihel culture and this double emulsion. because in these cases, the scaffolds of matrigel or another scaffolds, they produce different polarity. But with this, they found that there are random polarity, and in some cases, this polarity is a positive and negative, depends of the matrix. But in this double emulsion, they found that this polarity is random, but they don't find a difference between matrigel and this double emulsion, and they use another technique that is immunofluorescence in order to get what is this polarity, but they don't find also
1 minute left.
too much difference in these cases. For this reason, this is unclear. They don't find what is the polarity of this SI, and then with this they perform another experiment in order to see what happened with this culture with rocks, and they use the same schedule. After 3 days, they interrupt the spheroids. They erupted the emulsion, and they cultivate the spheroids with lung cancer cells, and they use drugs in order to see what is the ability of these cells. They use binaural bind, and this is the behavior of the viability in order to the concentration of the drugs, and this is the final result. Thank you very much.
Hello, everyone. You can see my screen.
Yes, Camilo.
Okay. In this occasion, I find the paper. It's very interesting for the title and reproduction of the large scales and bioreactor condition and the micro chips. This is important for in actually the cells in the industrial bioreactors are many strategies, but microfluidics is the powerful tool for produce on different production of for industrial or pharmaceutical, biological, and alimentary. Okay, and in this case, make a different models of chip for trap or that single cell, but for make them produce the different compounds. Okay, in bacteria. That's right. Produce in the figure 8 and show how the chip trap the cells in depend on the flow, and in the strategy it's capture a single cell, but on different wells of the more cells, right? After it's they can calculate the formulas, mathematical formulas for have Deloitte rates, the flow rates and the fluid for a trap single cell, and after analysis on produce different reaction into the micro device. After calculate on the signal of produce cell to cell, and after signal for microscopy or other strategies for takedown results. Okay, in the figure log, it may show the schematic chip on the capture the different cell of bacteria, and different chambers, and you can see after this fabrication of chips, the conventional method, similar we make in our laboratory. You can see in the figure 3 are how it's the channel, and after the cells inside and trapper in different chambers, right? Okay. After they analyze the different results, yeah, they can see the different reaction for positional of micro device. Okay, and in the micro device, you can see a reproduction different signals for single cells, or capture more cells into the chambers. Okay, it depends and calculate on level, vessel level for the producing the signal. For in this case, you can see the signal in the time of make a
1 minute left.
okay. In this case, you can see the different signal for the different part of a micro device. Okay. This in this occasion we show we look show a composites of the different signal, the frequency produce the reactive. Okay, and now conclude this strategy is improve the conventional method of its depend the time, and in the future this method for improve different industrial bioreactors or replace bioreactor industry. Okay, that is all. Thanks for the attention.
Hello, everybody. I present this paper called Modeling Other Minds, Bayesian Inference Explained Human Choice in Group Decision Making. This paper is the theory of machine learning algorithms such as reinforcement learning, where the authors present the social and psychological theory in order to model the way of humans make choices. Normally, humans depends the choices in order to increase the reward from actual state. The principal parameter of this choice depends on uncertainty. So this behavior can be modeled with many methods, in this case, according to a belief-based decision or belief-free decision. So in this case, Bayesian framework is used because the choices depend of the actual state and future states. In reinforcement learning algorithms, for example, the reward depends of the action choice in the present state and the reward in the future. So the experiments in this paper, the authors choose a game called PGG, where it's the standard in economy, where the people can be divided in groups, and they choose two different actions in order to establish a success or failure process. So in this case, depends the reward obtained of this choice can be positive or negative. In this figure, for example, the groups are joined in two states, for example, with 2 people and 4 people. The contribution probability is more easy than, for 2 people in a group than for 4 people in a group. This is because the modeling of the decision of group of 2 people is more easy to 4 people, because the decision depends the very different parameters. For example, in the figure E, the modeling, the linear regression of this behavior is easy. The modeling of with chi is equal to. The way of modeling this behavior is according to the binomial distribution, where the symbol theta is according to the uncertainty. So this can be modeling with the binomial distribution, but using the Bayesian inference. So in this case, the modeling of reward depends the probability of choice, different actions, and values of reward. In this figure, the author presents the set of all belief states, and in color yellow, the optimal actions can be choice with the people.
1 minute left.
Okay. Finally, this approach is compared with reinforcement learning algorithms such as school-learning, and the results can be described increase the accuracy of the predictions of new choices. So after finally this research, the author presents the improve the co-learning algorithm using this Bayesian inference approach. So in this case, the Markov decision process is more accuracy when the distribution probability is using according to the binomial and Bayesian inference calculate. So finally, this paper in general use the weight of group can be choice new actions in order to obtain the more reward, which is used in machine learning algorithms such as reinforcement learning. Thank you.
Good morning, everyone. Today, I would like to show you a paper about behaviors of glioblastoma cells in vitro microenvironments. The aim of this study is to understand the metastasis process of these glioblastoma cells, which produce brain tumor. So the idea of these authors is to mimic the microenvironment and replacing the extracellular matrix with collagen, because this hydrogel has the mechanical properties of the ECM. Okay, so here you can see the device that they use, and I would like to show you also in the supplementary information, they have the description of how this device is made. So they use photolithography to produce the PDMS stamps, and then coat the surface with fibronectin, which is represented in blue for the PDMS stamp. And then they also fabricate this other PDMS stamp, and they functionalize it with BSA, as is represented here in green. After that, they align the two PDMS stamps, and they fill these with collagen. So after the gel is formed, then you hear that the glioma cells are located in these spaces, in these wells. So let's see how the cells look when they start growing. So here you see that the cells are located in each of these wells, and then the cells that are not inside the wells are washed out with PBS. Okay, so here we can see how the different cell lines, they use 4 cell lines of glioblastoma, and worth mentioning, in ROFO they use LN229. And here you can see how they look in the Petri dish. So here, let's return to the paper. The idea is to characterize the morphology of these cells. So you can see here in these, here are in the columns, sorry, in the rows, you see the days of culture, and in the columns, you see the cell lines, the different cell lines. So here you see how they start growing, and how the morphology changes while the time passes. I also would like to show you this video of how the cells move, so that the authors can measure and characterize the way they invade, they proliferate, and also how they form spheres. Like in the case of the U87, you can see that they form neurospheres, and they are very good for invading. So they have these protrusions that helps them to migrate through the collagen matrix and invade the tissues. So here, with the fluorescence, when they normalize this integrated intensity of the fluorescence, here you can see the 4 cell lines and how they start growing. So here we see the proliferation rate, and also the authors normalize the cell area, and they report. And here is very interesting that they look at what happens in day 3, for example. Through the intensity, we know that the cell in U87 is the one that proliferates more. But when we measure the area, other things show. Here in the day 3, the U87 looks like it hasn't proliferated. So that's why they contrast these 2 parameters, the area and the fluorescence intensity. And they see that it differs what it's reported. So they attribute this to the different shapes of the cell lines. Here we see also how they measure the speed of the cells, and also these new parameters that we can look at. For example, the directionality radio, which is the ratio between the displacement and the length of the trajectory. So
1 minute left.
Thank you, Andrea. And here we see the area that a cell can explore. The average area, here is this parameter, it's the mean square displacement. So here it's reported, it represents the area that a cell can explore. And here we have also, they measure the protrusions. So here, these enable them to classify the cells. And here we see, for example, this cell line, the blue one, the LN219, it didn't show protrusions during this time. That's why here we see only the measurements of the 3 others. The average length of these protrusions, and these parameters help them to classify them into the cluster invasion cells, U83, the N220, it's expansive, it proliferates, it has a proliferation rate that is large, and also this SNB19, which invades, but in an individual way. Okay, thank you very much.
Can you see my screen?
Yes.
Yes.
Thank you very much. Good morning, everyone. My paper is tituled Bistability, Bifurcation, and Waddington's Epigenetic Landscape. First, I need to explain the concept of bistability and bifurcation briefly with this picture here. In physics, one simple model is the harmonic oscillation. The harmonic oscillation of the box of mass m can be modeled with a potential function and a particle confined to the well oscillating around the origin. Here, this movement is here, around the origin. Bistability is when the potential has two wells and the particle can oscillate in two different potential wells, here or here. Bifurcation occurs in these figures, for example, at the origin here. We have an unstable bifurcation point in this origin, and the particle can go to the left or right side. The particle has two possibilities to move. Returning to the paper, bistability and bifurcation can be used in biological systems by means Waddington's epigenetic landscape. Epigenetic landscape represent how a cell progresses from the undifferentiated state to one of a number of distinct differentiated cells phases. During development, the cells, the epigenetic landscape is in these figures. Figures 1. The cells is represented for a ball which move to towards the front of the landscape by means that lines. The right X represents phenotype or single time dependence state variable. From the X axis to the Y axis is the times, and the vertical zeta axis represents the potential function. Potential function is the determinate both the existence of the genes, proteins, and metabolites in biology. The aims of the paper is to answer the question whether this epigenetic landscape approach really describes what happens in the cellular system it studies. Is this approach completely? Okay.
1 minute left.
Okay. Two phenomena are studied here: the self-fade induction and the lateral inhibition. In the self-fade induction, the models is describe the evolution of the cell toward indifferentiate state. The author proposed one model, one mathematical model based in the Hill's function. And the potential function of these systems is that landscape, which is very different to the Waddington landscape. The difference is that the bifurcation point, in this case, is toward to the valley, to the settled node point. In the Waddington's landscape, the bifurcation is a pitchfork point, picture bifurcation point. That difference is important, very important for describe the biological system. For example, in vulval induction in C. elegans lab, for example. Conclusion: the bifurcation observed are different. The landscape in Waddington's landscape to the settled noded landscape. In this case, therefore, the biological process can be better understood by seeing the topological difference in the potential function of the epigenetic landscape. Topological difference in that function. Thank you very much.
Okay, any question?
Yes, I have a question. Gustavo, in this paper, what for strategy for the